Okay, I want you to stand up, stretch a little, and stand up, stretch a little, please, for me. The last speaker, poor last speaker who woke up at 4:30 a.m. Stretch a little and turn to the person closest to you, behind or beside you, and look him or her in the eye, and think about, think about that person a little bit, and then shake hands and say whether you prefer butter or margarine. Butter or margarine? All right, butter or margarine? That's a really important question because it's going to determine whether your marriage is going to work or not. Okay, and if you looked at this graph, there's a really tight relationship between margarine consumption and divorce rates, in Maine at least. This is not Maine, but okay. The point I'm trying to make is this is a ridiculous example, but there are two lines there, and they're really, really, tightly correlated. If you did any standard regression type thing, you think that there is a relationship between those things. But these are time series, and we all know that time series are very likely to be autocorrelated. There are lots of problems. Okay, so that's margarine divorce rates, and these are things we think we at least understand quite a lot about, right? But we're dealing with much more complex things. We're interested in diversity through time, and when we're dealing with wiggles like that through time, how much more difficult it is to think about what's driving these patterns, right? And we know by hearing the talks that have gone on today already, that these patterns are really produced by very, very complex processes that, well, can we ever actually hope to understand any of them and what actually drives these patterns through time? Okay, so apologies as well. I'm like Natalie, I'm not a real paleontologist, come from a biology background. And I would say that biologists are sometimes very lucky because they can go out and study biotic interactions like that. You can collect a lot of data. And if anyone would care, you could actually look at biotic interactions in my body right now. There are like bacteria and viruses doing things, and if anyone would care to measure it, you would get patterns like that, but you actually have some idea about the mechanisms that would drive these patterns, and even in a simple system like this is a very famous one that everybody would have heard about. It turns out that something as simple as a two-species system is actually highly complex. So do we have any hope as paleontologists? Okay, so that's sort of the motivation for the first part of my talk. Motivation being that we're interested in trying to at least get at some kind of understanding of biotic interactions that take place over a very, very long time scale of what we can observe in the fossil record. But the problem is that we know, as Julia already said, correlation is not causation, so what do we do about that? Time series data are not independent, and then there are all kinds of unmeasured things that we can never measure from the fossil record or anywhere else, but that we know might be happening, things that might be happening. And hopefully I'll have time to talk about something else, which is sort of related to the first, because the first part of my talk is it's going to be about modeling things we can hardly measure, and it's very unsatisfactory. So the second part of my talk is actually going to be about a real solid, real system, which you can touch and see. Okay, so that's in 15 minutes. Okay, sorry, that went a little too fast. Okay, so again, this is a motivational slide. What we have are processes that are translated into observations of some kind that you have in your computers. And from those observations, we somehow have to squeeze out temporal patterns that are proxies for something that we might be interested in. And for this part of the talk, we're going to talk about proxies for responses to biotic interactions, which is kind of vague, but I'll get there. And then from these wiggly things, what we want to have is an understanding of the system, whatever the system might be. So we're going to actually just focus on this arrow here and ignore the rest of it for the talk. And just as a little advertisement, Dr. Hannesdahl and I are writing a sort of review type paper for paleontology. It's almost done. That will summarize some of the things I'm going to be talking about here. Okay, so what I'm going to present right now for the next, hopefully just 10 minutes, is a tool. There are many tools out there, but I want to present a tool that we can use to model causal drivers in the fossil record. That sounds a little bit scary, but I'll just walk you through it. So the kind of causality I'm talking about here is a very specific kind of causality. It's called Granger causality, and it's not new, it's old. But there are a couple of features, which is basically these, and you may not agree with it, but this is Granger causality, and that's what it is, okay? So for something to happen, the driver has to happen before the response, that's one thing. And then the driver has to contain unique information about the thing that is being driven. And that's it for the original Granger causality, and there's been lots of expansions from the original 1969 paper. Okay. So this is the way we're going to do it. So this is a simulated time series. You can imagine it to be diversification rates or something, or phenotypic change, if you like, moving through time like this, wiggling up and down. And basically what we're going to do is to use linear stochastic differential equations. It's a mouthful, but I'm going to show you what it does. Basically, it describes this wiggly curve by saying there is an expected value. There is a stationary standard deviation. It's the size of the wiggles up and down. And then there's a half-life, which is half the time it takes for the wiggles to get back to that average value. So that's a basic linear stochastic differential equation that you can expand on. But it looks like this. Just ignore the way it looks. But all the elements are in there, in that equation. Okay, and it will be very familiar to many of you as an OU process that David Pauly just talked about very briefly. So it's nothing more than that as a basic equation. Okay, so that's our basic linear stochastic differential equation. And you're sitting there wondering, how do I use this to infer causal processes from the fossil record? Okay, so just very quickly, this linear SDE has a deterministic part. It's got a stochastic part. If you take away the deterministic part, you're boiled down to a Brownian motion. Description of Brownian motion. Okay, so let's just think about a driver time series and a response time series now. Then we have x1. Let's say that is your driver, or let's say this is environment, for example, temperature change, if you like. And then we have another time series, x2. Let's say that is diversification or phenotypic change. And you want to model the correlation between temperature and phenotypic change, for example. And so very simply, what this does is to introduce a term called rho in the stochastic part of the equation to measure the amount of correlation between those two time series that you're interested in. If you wanted to model explicitly a causal relationship rather than just a correlation, what you want to do is to manipulate the deterministic part of the equation. And what you can see here is that there's another term called beta, but now x1 is embedded in the deterministic part of x2. So when x1 changes, x2 is going to follow, okay? So it's really as simple as that. Okay, so linear SDEs as a time series tool is that the great thing is that it differentiates very explicitly causal versus Granger causal versus correlative relationships. The great thing for us as paleontologists is that it doesn't require that your measurements are made at the same time. You don't have to forcefully bin your data or anything like that because it's a process. So we don't need that as long as there's some overlap. You can't have one time series happening in the Devonian and then another time series happening in the Cenozoic, for example. There has to be some overlap, but you don't need measurements to be made at the same time. We can accommodate uncertainty in your measurements. There's plenty of uncertainty in everything we estimate from the fossil record, so it's great that it does that. And it can also find causal connections when you have latent drivers hidden underneath. And that sounds a little bit mysterious, it's like black magic, but it really isn't. So let's say this is your measured time series, and the blue data are not available. You don't see it. But it turns out that the autocorrelation is of with influence and without influence from the blue time series that you don't see and you don't measure are different. So there's actually signal from your measured time series that will tell you that there's something underlying the process. Okay, so these are the really good things about linear SDEs. Okay, then you're sitting there and thinking, oh, that sounds really horrible and complex. Why couldn't you just use simpler methods that would do the same job, right? I don't like doing complex things. I would prefer to do simple things. So if you wanted to look at Granger causality, why don't you just like lag your driver series, not the driver, but the response series, and then you have a Granger driver, right? So you could do that. The other thing that I've mentioned before now is that time series data are not independent. If they're not independent, why don't you just do first differencing or something to remove that autocorrelation? So I wanted to convince myself that I'm not being stupid by wasting a lot of time like learning new methods or struggling with difficult things when simpler methods will do the job, right? Because we're interested in biological processes, we're interested in paleontology, so as long as we're able to resolve our issues, it's fine. So we did some simulations, and basically what we did is we had two time series, X and Y, and then we modeled those three relationships between them, like no relationship, a correlative relationship, just like the butter, margarine, and divorce rates, and then a relationship where it's actually causal, something's driving something else. We did the simulations, and then we applied SDEs. We did regression, like regular regression, where you have, you're something that's regressed on something else, and then we did first differencing and then regression to remove the temporal correlation, and to model a causal relationship, we just lag the response time series. Okay, so you're going to see this horrible table, and I'm just going to try to guide you through it quite quickly. So in orange or yellow are the truth, and these are the inferred relationships using the three different methods that I just talked about. So blue is the one I'm trying to promote. It does quite well. If you just did regression, you don't hit the right answer very often. If you first difference and then regress, it is a little bit better in some cases, but not a lot. But the point is that we do get a lot of false positives and false negatives if you read this way and that way as well. So I've at least managed to convince myself that I cannot rely on, just lagging my time series in order to get a causal relationships. Okay, so that doesn't work. Okay, so now back to biotic interactions, which is what I'm supposed to be talking about. A long, long time ago, before some of you were born, maybe, Gould and Callaway put together this data, and they convinced themselves that there's basically no relationship between the diversity of brachiopods and the diversity of bivalves. A lot of people have been saying that bivalves have driven brachiopods to extinction or almost extinction, but they disagreed, and they put together lots of data and sort of blame it on this extinction, right? So we were interested in looking at this question again, but using linear SDEs to model the relationship, which may or may not be causal. So what we did, so Trond Reitan is a statistician, and Paul Harnick, a good colleague of mine in the US. What we did was we took paleobiology database data. We estimated the extinction and origination rates of brachiopods and bivalves, and we took care of the varying sampling through time as well. And then we looked at those 4 time series and the relationships between those 4 time series, and basically we convinced ourselves at least that there actually is a relationship between bivalve and brachiopod diversification, where when bivalve extinction rates are really high, brachipod origination rates are high. So there's sort of a release. When the bivalves go away, the brachiopods are able to come in now and then. Okay, so that was just one case study of how you could use these causal inference tools as applied to paleontological data. But that's not very satisfying because, you know, this is like data somebody else collected, and it's global data. We don't really know what's happening locally, but when we do think about biotic interactions, what we really want is to see things interacting and having an effect on the guy you're interacting with, right? So, and enter the world of bryozoans. Bryozoans are marine colonial organisms, and they're great because they preserve in huge quantities, and you can actually look at the interactions preserved in the fossil record as well as you can in when you're diving, for example, if you had a microscope with you while you're diving. They're very small. In any case, here what you can see in these beautiful SEMs taken by Paul Taylor and Emanuela Di Martino, who's sitting here in that corner, are different bryozoan species that are competing with each other for space and trying to take, you know, the neighbor's space, because without space you cannot eat and you cannot reproduce. And we can see that frozen in time. Okay, so the data I'm going to be presenting to you, I know this is supposed to be a modeling session, but I'm always motivated by real data. So let's go through the real data first, and then we'll do some modeling on it. Okay. So here's where we've been. It's very well characterized geologically. We know a lot about the stratigraphy. Everything has been worked out. Even the bryozoan taxonomy has been quite well worked out in this part of New Zealand. When we're talking about 2.3 million years of sediments, where you can look at these bryozoans interacting with each other frozen in time. And we also have data from contemporary samples that are equivalent. And we've been out there doing the fieldwork ourselves, so we actually, I think we actually understand the data quite well. So we've been out there twice, but the results I'm going to be presenting to you are actually just results from our first trip, and there's just lots of data in the lab, and we haven't looked at them yet because we're just oozing with bryozoans. Okay, so first, just the basic data that we have. So this is a shell. It's 2 million years old, approximately, and you can see with your naked eyes that there are different bryozoan species, and they're growing on top of each other. Those very tiny words there, we can actually identify most of them down to species level. And most of this work has been done by Emanuela Di Martino, and she's trained one of our master students who has also contributed to a lot of the data that we're going to see in a bit. So basically, when we talk about biotic interactions between bryozoan species, what we're talking about are spatial overgrowth, where they're competing for space. So you can have a win-lose situation where one colony grows over the other. It can be different species. It can be the same species. And then there's a draw situation. There are two draw situations, actually, where you sort of grow towards each other, and then you meet this other guy, and this other guy is about as strong as you just stop. And then there's also this thing called reciprocal overgrowth, where in one part of the colony, A overgrows B, and in the other part of the colony, I'm not sure if you can see my fingers, but it crisscrosses and the other guy wins. Okay, so that's the basic data. We have species, identities, and we know who's overgrowing whom, and it's over to 2.3 million years. Okay, so that's a real picture of what's happening, illustrated in these SEMs. Okay, so what I want to show you now is work that we're currently doing. We're sort of trying to wrap up the work now. So a very simple question: what's a winning bryozoan? Who wins? Right? So in some previous work we've done already, so we've described this system, and we've looked at the competition amongst these bryozoans colonies. And what we think we know from this previous work is that it's really good to be big. And big not in the sense that the colony is big, but the individual modules or zooids. So like I said, this is colonial organism, and each unit is produced by the same genetic information, but it's one colony. It's like a plant kind of. Okay, so if you are large, that is your zooids are large, your modules are large, you tend to win. Okay, so bigger is better. Okay, but if you had a weapon in your hand, is that going to help you? Oh, there we go. Shouldn't use the Mac. Okay, that was supposed to be a dagger. So if you're a large guy, are you going to be afraid if a little guy points a dagger at you? I'm glad you're still entertained. And the other question I want to answer in this talk is whether time matters, right? So we've got 2.3 million years. I think it's going to be changing over time, or is it the same set of traits through time? Okay, so does time matter? Okay. So let's talk a little bit about the traits because they matter. So we have things like defense traits, they look like defense traits, we don't really know. Okay, there are spines, you can see some spines here. I would think that if the spines are pointing towards you, it's going to be really hard to like hop over the guy with spines, right? And then there are these things called avicularia. There are many proposed functions, but then we know that some of them sort of like do this to like little worms and micropredators. So the question is, if you had these things, are they going to like deplete your energy so you have no time to grow or no energy to overgrow your neighbor because you're stepping away at micropredators? Some other traits, the kind of shield you have over your body, whether you're kind of a naked bryozoan or you're like fully shielded, like a Death Star. And are you a very common bryozoan? Are you a rare bryozoan? And we've already talked about zooid size. So there are a lot of traits you can look at, and the question is, what combination of traits makes a good, strong, competitive bryozoan is the question. Okay, so that's the question. We'll answer the question of what describes a good New Zealand, which is where we've been, a good New Zealand winner. Does time matter? And unfortunately, I'm not going to have time to talk about this, but we were really interested in trying to predict completely novel systems, whether we could use like traits from New Zealand to predict completely novel systems. So I'm happy to talk to anyone about that afterwards. Okay, so this is the modeling bit. It's not going to be very long. But basically, we have discrete outcomes. So as colony A, let's say I'm colony A, I have the choice of meeting you and winning, or we can stop, or you can overgrow, right? So it's an ordered response, and it's discrete. You either win or you don't win, right? So we're going to do ordinal regression, but we use this model called the cumulative logit model, which is actually quite standard, but there were some funny things that we had to deal with. So basically, that's what the model looks like. It's got a logit function. This is such that we can actually model the traits in a linear fashion. So those are the ordered outcomes. In our case, there are 3 outcomes. And the X actually is the difference of the traits between the 2 bryozoans. So like you and me, glasses, no glasses, long hair, no long hair. And sometimes we might be the same, like long hair, right? Okay, so that's the difference in traits, and that's just, the logit is just log of odds, okay? So what I want to remind you a little bit is that we actually have a lot of traits. I've shown you a few here. You can break them down in different ways. So because we have so many traits and we're interested in trait-trait interaction, like figuring out whether it helps to point a dagger at an elephant or not, it turns out that we really have a lot of models to search through. It's crazy. So we had to devise some crazy search algorithms to find the best models to describe our data. But let's just skip over that and answer the questions, because it's almost beer time. Okay, so basically what I want to show you is that in the next slide, what we're focusing on is the beta that is being estimated. It's labeled as estimate. And that's going to tell you whether the trait matters or not, and in which direction, okay? Okay, so this is like one of Natalie's slides where we just have the data. I haven't had time to make it pretty. But in any case, what I do want to share with you is that these are the beta estimates. And let's just walk through the important ones. Is time important? Yes, time is important for one of the traits, and the trait that is important for is size. So if you looked at the estimates of the beta here, they're all positive, which means that bigger is always better. But it's not always better to the same extent. You see quite a lot of variation in how much better it is. Sometimes the effect is not very big at all. But size is an important trait, and it actually explains a lot of the variation in the data. I was like, okay, so size is still important, and time is important for size. Do we see any effect of weapons versus interacting with size? There is no such interaction in here at all, and we haven't seen that in all the other versions of the best model we've looked at. So there isn't that, mouse holding a dagger effect. And another very strong effect that we've seen is actually the way the bryozoan grows. So most of them are quite flat. They grow in a single zooidal layer, but some of them, they're able to make these multi-layer stories, which makes it perhaps difficult for someone who is quite flat to jump over or to grow over. So that effect we see quite strongly as well. It's this one in green. So this is one of the best models that we've found because there are many different ways to describe what is best. I don't have time to go into that right now because it's almost beer time. But I just want to end with a little advertisement for my lab. So I've been working quite a bit on bryozoans the last couple of years, and we're going to work even more on bryozoans, and there are lots of colleagues here in London who are in this with me. And we look forward to telling you much more about bryozoans in the next few years. There are a couple of posters and a bryozoan talk on Tuesday by Emanuele Di Martino as well, so please come for that. Yes, that's the end. Thank you.